Dr. Taruna Madan from National Institute for Research in Reproductive and Child Health in Mumbai. She is currently holding the post of scientist staff at head of the innate immunity department in the institute. She has completed M.Pharm and PhD from the DIPSER and Institute of Genomics and Integrative Biology in 1999. She has more than 20 years of the professional experience. And she was awarded as a Young Woman Bioscientist Award in 2004 by Indian Science Congress, Chandigarh. And she had received Indo-US Christman WIS Fellowship in 2019. So I called Dr. Taruna Madan to deliver the lecture on recombinant human innate immune proteins for therapy of prostate cancer.
Thank you so much, sir. And a pleasure to be here in this meeting, and thank my dear friend, Professor Sham and Professor Bansal, who have invited me to be here to share some of our findings on a molecule that is expressed by the human host. And Chandigarh has been a great city, and I love this city, and this is my nth visit to Chandigarh. I can't even count how many times I've been here, but mostly came to collaborate with PGI for our immunodiagnostic kit. I'm not sharing that data today, but sure, in due course of time and further meetings, we will talk about that. So as Professor Sani introduced, recombinant human surfactant protein D, a novel biologic for immunotherapy of cancer. I will be focusing on our data on prostate cancer, which is what we have worked on, but will share some of the findings by other groups on other cancers. So, surfactant protein D by structure is a C-type lectin. So lectin, we all understand that it is a carbohydrate recognition binding protein. It has specificities towards sugars, or to say more specifically, the surfactant protein D, sorry. Okay. What's happening? Why it's not going back? Can I? Yeah, started. Thank you. So what I wanted to show you here, this is the molecule that we are working on, surfactant protein D. It's in the group 3, and the family is known as collectin. And why it is called collectin is because it has these long collagen domains. These are gly-XY repeats, and they coil together to form a trimer, and this molecule is a tetramer of trimers, so 12 molecules together. And what we work on is, I'll share with you, is this small portion which we have generated recombinantly and has all the functional capabilities of surfactant protein D, other than its collagen domain properties that are to activate the immune system. So discovery of collectins. So I pay my gratitude to Professor K.V.M. Reed, Fellow of Royal Society and former Director, MRC Immunochemistry Unit, Oxford University, with whom I did my postdoc. And that is from where I took this work forward. So Professor Reed's group had purified, characterized, and did cDNA cloning of human lung surfactant protein D. The mean normal serum concentrations of SPD ranges between 48.7 and 109 nanograms per mil. Physiological levels of the other 2 collectins are also showed here. So these are the 3 collectins that are part of the collectin family. So here we show you how the structure formation happens. So you have these 3 carbohydrate recognition domains: trimerization, oligomerization, And when you have tetrameric oligomerization, you get SPD. And now with the electron microscopy structures, we know that this oligomerization even goes further, and you have a molecule which resembles a wheel with spokes, almost like our Ashok Chakra. So here you can see the highly oligomerized electron microscopy structure of surfactant protein D. Here we call it as fuzzy ball, as named by the discoverer of the structure. So pattern recognition, this is a pattern recognition molecule. It happens through carbohydrate recognition domain. It inhibits an array of pathogenic bacteria, fungi, and viruses. And that's how I started working on this molecule, because we were working on a fungal infection, pulmonary fungal infection called aspergillosis. It inhibits T cell responses and restores quiescence in activated lymphocytes and monocytes. I'm sure you must be, having that this particular property is so important for COVID infection, that it inhibits T cell responses and restores quiescence in activated lymphocytes and monocytes, which is what is needed in most of the chronic inflammatory diseases, not only COVID-19, that you need to bring down your immune system to the normal, or we need to restore the homeostasis. Enhances phagocytotic clearance of apoptotic necrotic cell, which is another need of the hour. Systemic and tissue expression is altered in infection, inflammation, and cancer. So this is the kind of receptors. There are plethora of receptors that are known to bind to collectins, and that is how these proteins play an important role in immunoregulation. I'll not go into detail, but I'll tell you that the NF-kappa B pathway is the major pathway that is affected by these receptors. Once these proteins are occupying, the collectins are occupying these receptors, the pathogens are not able to trigger the pro-inflammatory pathways through various pattern recognition receptors. And these are soluble pattern recognition receptors, collectins, whereas you know that toll-like receptors are membrane-bound pattern recognition receptors. So we tried to understand the regulation of SPD expression, and we knew that dexamethasone and insulin induce SPD expression in the lung adenocarcinoma cell line. We were working in the reproductive tract. As you know, my affiliation is with ICMR's National Institute for Research in Reproductive and Child Health. So we tried to understand the presence of this protein in the reproductive tract and its regulation. And you see here that the estrogen regulates SPD production in the murine uterus. And there is a synergy when you use estrogen followed by progesterone, which is what happens in the normal menstrual cycle. And if you see the male reproductive tract, you see here the testosterone also upregulates. So there is a developmental regulation of SPD in the male reproductive tract. So to decipher the role of these pattern recognition proteins is our main aim of the research. We have worked of its role in HIV/AIDS. We are working for its role in pregnancy and fertility. We are also working in their role in prostate cancer and breast cancer. And today I would be sharing only the prostate cancer data for diagnostics, therapeutic applications. Dr. Uday Kishore is Director, CIIDM, Brunel University, London, UK, and now he has moved to Imperial College, London. And he is the person who had generated recombinant fragment of human surfactant protein D. And this is the structure of the protein. So we are taking only this neck region and the carbohydrate recognition domain, or the C-type lectin domain as mentioned here. So this is how it looks like, and this is the crystal structure solved of recombinant fragment of human surfactant protein D, and this has these many amino acids. So it's a small protein of 17 to 18 kilodalton, but on SDS page shows around 20 kilodalton band. So this discovery of this protein's role in cancer was in serendipity, and the credit goes to my student, graduate student, Dr. Lakshna Mahajan, who is now teaching in Swami Shraddhanand College in Delhi. And what we worked on is, we were working on eosinophilic interaction of surfactant protein D. And what we found was that the eosinophils which were coming from allergic asthmatics were undergoing apoptosis. Sorry about that tongue twister. So so we were perplexed that why it is, and to the healthy eosinophils, it was not affecting. Then we got an eosinophilic leukemic cell line from our collaborators in US, and we saw that the cancer cells were put into apoptosis. So it showed us that whatever cells are in activated stage or in proliferating state, they are put into apoptosis. And we confirmed that when we are taking healthy PBMCs, there is no effect on the viability, whereas when we use Jurkat, Raji, THP1, MCF7 cell lines, we found that the viability is significantly affected, suggesting that there is an anti-cancer role in this molecule. And this is the eosinophilic leukemic data, which we found that it is dose and time dependent manner. RFHSPD is inhibiting the viability. SPD inhibited survival of A549 human lung adenocarcinoma cells by reducing EGF binding to EGFR. So after we discovered that, soon, just next year only, Hasegawa et al. Showed this wonderful study where they found that SPD has an ability to bind to the epidermal growth factor receptor, which is very, very critical to the cancer cell survival. So once SPD is blocking the EGF-EGFR interaction, it is inducing apoptosis in the cells, was their hypothesis, and they proved it in the lung adenocarcinoma cells. So prostate is the second leading site of cancer among males. That's how we started working on prostate cancer, and definitely has similar kind of incidence in various Indian cities. Radiotherapy, we all know, is used for killing cancer cells in various cancers, including prostate cancer. Chemotherapy that is used for prostate cancer involves docetaxel, methoxantrone, cabazitaxel, and istramastine. Prostatectomy is another solution that is offered to the patients where the prostate is removed, and you have orchiectomy where you have testis removal, so that you have, we call it as androgen deprivation therapy, because it is understood that androgen fuels the prostate cells to proliferate, and therefore it was discovered that if we are depriving them of testosterone, that can help them to survive longer and tumor-free progression. But the thing is that very soon it was realized that all these patients who are undergoing androgen deprivation therapy, they are developing androgen-resistant form of prostate cancer, which is even more deadly and metastatic. Immunotherapy, we all know that there are vaccines that target prostate cancer antigens, priming the dendritic cells and of the patient itself, and we have this clinical immunotherapy program, and it is called Prostavac PSA and Sipuleucel PAP, which is the prostate acid phosphatase, which is a specific molecule produced by prostate. And prostate-specific antigen, we know that it is one of the diagnostic biomarker for prostate cancer. Oncolytic virus therapy is also in clinical use, and disabled virus is targeted to tumor cells with the gene delivery and followed by anti-herpes drug, and this has shown some promises. Checkpoint inhibitors, as we all know, they block the inhibitory molecules that are present on the tumor cells so that the immune response can be activated against the tumor cells. Your own host immune response can help you in overcoming the cancer. Adoptive cell therapy, removal and modifying T cells from the cancer patients, and again reintroduced to improve immune system and anti-cancer response. We call it as CAR T cell therapy, if you happen to know. And Dr. Rahul Purwar is doing clinical trials for various cancers in association with Tata Memorial Hospital in Mumbai. The issue is that adaptive immunotherapeutic approaches remain unsuccessful. Only 15% of the cases, whatever cancer it may be, are responsive to these immunosuppressive therapies or immunotherapeutic approaches. So because we have a cold metastatic prostate tumor microenvironment that does not let these immunotherapies work. So the important target is to remove this immunosuppressive cold prostate tumor microenvironment. And induction of tumor cell apoptosis by the intrinsic cellular immunity in the tumor microenvironment. These are the two things that are gaining momentum. And you must have heard about recently a clinical trial in cancer patients that showed 100% success. So how they did that was they identified what are the markers to stratify the cancer patients, to identify those 15% who are going to respond to these immune checkpoint therapies, and that's how they identified those 15% and gave their therapy and showed a 100% response. So the important thing is, what do we do for the rest of 85%? So expression of SPD was reported in prostate way back in 2000 in human prostate tissue as a protein level. You can see the prostate tissue is expressing surfactant protein D. The brown staining is surfactant protein D. Surfactant protein D expression in prostate cancer was discovered or reported by Kankavi et al. In 2013. And these two panels, A and C, are from the prostate tumor tissue, and rest of them are from the healthy prostate gland. And you can see not only the expression level has gone up, but the localization of the protein has also been altered. So on the basis of this, my graduate student, Gargi Thakur, now research associate with High Media Laboratories and now moved on to Tata Memorial Hospital, the prostate cancer group, for further research. So she discovered the anti-cancer role of RFH-SPD and identified its potential receptors on prostate cancer cells as well as multiple mechanisms. So I'll not go into the detail. What we show here is that the biopsy tissues which were taken from Tata Memorial Hospital group, prostate cancer group, showed enhanced apoptosis when they were treated ex vivo with recombinant fragment of human surfactant protein D. And these were the various mechanisms that we found out how intrinsic pathway of apoptosis is triggered by RFH-SPD exogenous supplementation to the prostate cancer cells. SPD increases karyorrhexis and membrane bleb formation in Jurkat T cells was reported after we reported the prostate cancer cells. This paper came in 2017, and we also confirmed that LNCAP and PC3 cells. So this is androgen-dependent prostate cancer cell, and this is androgen-independent prostate cancer cells. We took 2 cell lines to confirm that RFH-SPD has a broad spectrum of activity. In view of interaction of SPD via its homotrimeric CRD region with various ligands as well as receptors, we tried to assess the membrane interactome of this molecule on PC3 cells using proteomics. And this is what we got with help from Dr. Susan Thomas, who is heading our Biomedical Informatics Center. And here you see HSPA8, which is also known as GRP78, or glucose regulatory protein 78. This molecule is a chaperone. This is present inside the cytoplasm and endoplasmic reticulum to shape your molecules, their structures in proper form. In the cancer cells, GRP78 does moonlighting, moves over to the membrane, and is doing some different functions which lead to its oncogenic or tumor promoter role. And here we found that surfactant protein D is binding to HSPA5 or the GRP78. And this is the bioinformatics analysis which showed us what are the amino acids that are involved in this interaction of RFHSPD and GRP78. We also confirmed using ELISA that this interaction is indeed taking place. Regarding the regulation, we found that there are 10 androgen responsive elements in the human SPD promoter, because we had found that testosterone upregulates the expression of human SPD in the prostate gland, and that happens to the androgen responsive elements that are present in the promoter region of SPD. We had shown dihydrotestosterone regulation of SPD transcript in the prostate cancer cells as well. So you can see here the androgen responsive cancer cells as well as the primary prostate epithelial cells. They show increased expression of surfactant protein D, and whereas the PC3, which is an androgen resistant cancer cell line or a metastatic prostate cancer cell line, did not show that, and the cell viability was not affected significantly with the DHT. So we moved on. Another graduate student, Ms. Kasturi Ganguly, joined us, and she is now deciphering the multiple mechanisms of innate immune surveillance in prostate cancer, and she's using the transgenic mouse model of prostate cancer. We call it as TRAMP mice, where you have a transgene which is regulated by probasin promoter, which is responsive to testosterone. So whenever the testosterone will come up in the mouse, male mouse, the prostate will start developing tumor. And this is the model that we are using. And so what she showed was that surfactant protein D is present in the early stage of tumor. It is there in the late stage of tumor. But when we are seeing on the Western blot, the protein is degraded. The protein is present, protein is synthesized, but the degraded protein is present because you have several enzymes coming up in the prostate tissue during the cancer progression, such as elastase, and we showed that the cleavage or the cleaved SPD is actually coming from the neutrophil elastase that is going into the prostate tumor. Treatment with recombinant fragment of human SPD and civilistat, which is a neutrophil elastase inhibitor, showed that M1 macrophage polarization can happen in TRAMP explant. Now, the M1 macrophages are anti-tumorigenic, and having them in the tumor tissue is showing us that it is really working through the immune activation. So it is what I said was the cold metastatic tumor microenvironment is now being converted into a hot microenvironment, which is anti-tumor. Induction of immunogenic cell death by RFHSPD in the TRAMP C2 cells and TRAMP explants. We had increased elastase producing immune suppressive cell in the tumor milieu, and we could show that this treatment is changing them. And as I mentioned, elastinolytic degradation is there. So we use the civilistat, which is an elastinolytic inhibitor, and that's how it is, the combined therapy worked for us. So this is what is our hypothesis, that immature DCs, M2s, neutrophils, and SPD transcription is inhibited. Important thing to note here is that if you give androgen deprivation therapy, your prostate cells are going to stop production of SPD. So although androgen deprivation therapy is helping you in some way, but it is inhibiting some of your anti-tumor host proteins that can help you. So here SPD transcription is inhibited. When we give RFHSPD along with civilistat, we saw that we have PMN MDSCs going up, M2 macrophages going down, M1 going up. We had mature DCs, and we had HMGB1 release, which is again another kind of pattern recognition receptor soluble form, and it attracts immune cells to the prostate tumor tissue. This is to summarize what all my lab has been doing to understand the role of collectins in human reproductive health, and it is not only the prostate tumor we are working on. We are working on fertility, female reproductive health, HIV, and we are working with clinical samples, patient cohorts, animal model, biopsy models, ex vivo, and we are now planning to move into the physiological systems, microphysiological systems. We call them as MPS, and that is going to be the next way of testing drugs in the pharmaceutical industry. And since COVID-19 was there, and this is a lung surfactant protein, we tried to understand the role of recombinant human surfactant protein D in COVID-19, and we found that it is very promising. We have patented it. We have got an international patent on it, and we are progressing further to produce formulations. So this is the basic molecule that we have worked on, a recombinant fragment of human surfactant protein D that we produce in mammalian CHO cells, and this is how the purified protein looks like. And we go for affinity chromatography because it is a lectin, so we can go for that. And the clone development pathway in CHO is in progress at the large scale with the help of a pharmaceutical company. I end here. The more I live, the more I learn. The more I learn, the more I realize, the less I know.
Thank you, Professor Madan, for the interesting and elaborative data on the collectin family. And very good data, I think so. And looking into the prostate physiology and current treatment options, are you thinking to combine this approach with the checkpoint inhibitors?
Yes, very appropriate question. Checkpoint inhibitors and RFHSPD can work in synergy because surfactant protein D is known to attract the immune cells, and that is what the principle is. And we have actually preliminary data on showing that some of these immune checkpoint inhibiting molecules that are present on the tumor cells, they are being downregulated by SPD treatment. Yes, sir.
Great. I think. Any questions from the audience? Yeah, please.
Yes, sir. Yes, sir.
We should do. Further, you should do. So, to taking into the clinics, because I see it's a 20 kilodalton protein.
Right.
Very big.
Very big. Very
it
big.
should be immunogenic.
Very big. But it is lesser than an antibody, right? Absolutely. It is an immune protein. It's a host protein. So we have already checked its acute toxicity. Although not in the intraperitoneal or intravenous route, since we were working off its relevance in the lung infections, we have worked its acute toxicity through the inhalation route, and we have seen that it has a very high toxic dose. So the therapeutic index that we have is higher than 10, so we can definitely use it. Yeah. Regarding macrophages, sir? Macrophages have different receptors, as I showed in my slides where I showed plethora of receptors. So macrophages do have those receptors, and this protein binds to macrophages and in fact enhances their phagocytotic capabilities. Yes, sir.
It's a nice presentation, ma'am.
Thank you.
I want to ask one thing. Just you have mentioned that it's targeting the checkpoints and it's targeting the EGFR also.
Right.
Is it targeting like any other kinase domains like serine kinases or like BRAF or other chaperones like HSP90, as you've mentioned about HSP90 as well? Like, is that a basic mechanistic action? Like, is it either targeting the chaperones or like serine threonine kinase or any other tyrosine kinases domain as well?
Very nice question. So EGFR has kinases downstream. So since it is binding to EGFR, it is not